Ladies and gentlemen, we're live with Matt Powers, who just kicked off his newest Kickstarter, Regenerative Soil Microscopy. Now, this is the second in a trilogy of cutting-edge permaculture books. And the first one we promoted a while ago. It was Regenerative Soil, and I have my copy right behind me. And the new one is going to be Regenerative Soil Microscopy. The Kickstarter just kicked off at 8 a.m. this morning, which is less than 12 hours ago. There's huge support, 23 backers. We need to get 25,000 is the goal. And Matt, welcome to the program.
Thank you so much for having me.
Yeah, indeed. Are there stretch goals?
Yeah, I have a lot of stretch goals because the reality is I'm doing something old and something new. I've done books before. This is very much a part two of a series of the most fertile area that I've ever really studied and kind of taught, the soil. But this is going to tie in with the database. And the thing is, the only way to actually evaluate and test the efficacy of testing of any sort of test is to do it against other tests, and then to collect all the tests and then look at them. And if we do this in a public, open source way, and then have the data play out, we're going to be able to see, like, oh, I'll see my soil, how it changes over time and track it. But you'll be able to compare it to the bioregion, to all the climate analogs across pHs or everything. And so suddenly we have the ability to see the future if we do change our soil. Let's say we like, oh, I want to have the exact same soil as I have now, except I want 20% organic matter. You can find someone with that soil in the database and then read up on like what their biology's like. And so you're like, okay, now I can grow compost that highlights that biology because I knew from someone else's test that they did a compost and this is the biology that maps out, and it's been done over 1,000 times. And so we know that's the way it works. So we're going to have new recipes. We're going to have new insights for all farmers. We're going to have new reference points. So you're going to be able to, as like a gardener or a farmer, know how you measure up to all the other soil in the area, and as well to yourself over time. So you can be like, oh no, I know what I'm doing. Let me show you. And you can show people, go check out my website, type in, you can make up your own username. It's going to be an open source. Everyone can upload into a community rated database. And it's going to be like a library that everyone can search it, but I'm also going to have DNA. So, and then allow people to do the raw video and pictures. And with the AI technology that's coming in for counting the microbes and identifying things for us, and then cross-referencing that with the DNA and using AI to go through all the DNA and compare that, we're going to be able to actually unlock things at a crazy next level. But we need to just fund that. You know what I mean? We need to be able to actually have people know how to do it. And that's why I wrote the book and the course. So people know how to use a microscope, how to pair with all these other testing methods. Because the reality is, it's like we can do salinity on our own. We can do NPK on our own. Yeah. We can do redox. I've got an ORP meter now, an oxidation reduction.
Pretty cheap.
The Philip Callahan meter, paramagnetism. But think about this for a second, everyone. What if we map the biology to like everything, to the time of day and the place on Earth? And suddenly you're like, oh, wait, moon cycles do have an effect on biology. And I'm not saying it does, but we would disprove it or prove it within 6 months.
Right. So this is next level stuff. If you're taking the first course, if you know about permaculture and you're out there and you're in your garden and you're putting in the beam, or you're using EM1 and you're chop and drop and you're doing cover crops and doing all those things to improve the soil, how do you know how good your soil is? And how does it compare? And do you know what's in there? Because literally in our permaculture orchard, it's now 5-6 years old in some areas. Some things do amazing and some things aren't grown so funky. And I need to know why. I mean, I'm doing the same thing everywhere. And so then I will research individual plants. I'm like, well, maybe this plant has a special need or something. No, that's not the problem. The problem is because of this, in this particular berm, there's some different soil chemistry going on, different than the berm next to it. Even though I was doing the same thing, something else is happening in the soil. And with regenerative soil microscopy, if you have a microscope and if you take this course, you're going to be able to take a thin section, a slide, put some solution in there and take a look at it. And will we be able to learn to find bacteria as well as fungi? I mean, because the ultimate goal here is to have a database where independent citizen scientists are providing the data to that database for the benefit of all humanity.
And also to develop our fluency. So like, let's say you have a nematode and you're like, the mouth parts look like a fungal feeder. Well, you type in fungal feeders into the search database and you get 40 or 100 different images, and you start looking through the images and you're like, oh wow, I definitely have that. And then you look at the other nematodes and see what they are, and you're like, I definitely don't have that. And so you get to create this visual fluency. And the only way that we're really going to figure out the next level of how everything really works is to interconnect it and to give everyone access.
Yeah.
And so I want to empower people with the tools. That's what the toolkit that we were talking about. I want to help them with the course. And the only other course that's like this is like $5,000. So this is an intensive course, but it's also a lifetime community. So it's not like a one and done kind of thing. You're part of this community, and we're helping each other and constantly learning and growing. And I teach people how to do experiments, how to create their own test protocols, because literally that's how like all the science is done. So it's like, if you don't teach those parts, it's like, what are we doing? You know what I mean? It's like, how are we going to actually get people to think critically about what we're even doing to actually push it forward so that we increase comprehension? So it's been, and what happened is this. I did DNA testing on the best compost I could find, Catalyst Bioamendments, and it came back with all the EM members. So all the EM guys and gals, all the microbes, here we go. They came back inside the compost in large numbers, and also rhizobia, which is kind of cool. So the nitrogen fixing beans, and our clover, it's already in the compost.
Yeah.
Boom.
Yeah. It should be in all perfect soils. The rhizobia should be present.
Yeah, that's what I'm finding. Mm-hmm. So what's so incredible is I did this DNA test. I saw that, hey, good compost is facultative, meaning we use aerobic composting, and things are aerobic as we're sterilizing it. Essentially, it's not truly sterilizing, but we're sterilizing it of pathogens for the most part. Though if we don't do it hot enough, we actually train Shigella, Salmonella, and pathogenic E. coli to live in that condition. So to be really careful, make our compost really hot, or finish it off with worms. At first, I didn't believe this was true, and I had to go research it. A student said this to me in one of my classes, and I was like, I must look this up, because they said that if you use earthworms in weeks, all the pathogenic E. coli is gone. And I was like, really? That sounds like magic. And then I looked it up, and it's true. And this is why Catalyst Bioamendments, among many, many other commercial composters, finish their compost piles with worms and several months of the worms working on it in a static pile.
Something in the excrement that's actually like a pesticide to the salmonella.
Yeah, all of it. And so this is like a miraculous breakthrough. And it's like something that, like, A, we should just all keep saying and spread and make common knowledge. But B, we're going to see all this play out. And if there's caveats for that, we'll see it too. And then we'll figure out, if we're doing thorough testing, we're going to see that, oh, it's, if we're deficient in these 3 elements, the microbes in the earthworms don't properly work, and then the enzymes, you know what I mean? Like, so we want to figure out those caveats, we want to figure out those conditions, and the only way we figure that out is by being honest and open, willing to be wrong, and entering this, like, this kind of community ranked and community solution sourced and really open and encouraging environment where people can go on and learn and grow and learn from others who are doing really, really good work. Because what'll happen is you'll have people who do unbelievable work, and they'll be higher ranked, and people will be like, okay, that's the standard, I can do that, and it'll lift everyone.
Yeah.
So
So let's talk about this, the lifetime access, because this is important. Anyone who's getting involved uh, in the Regenerative Soil Trilogy, specifically part 2, which is microscopy, if you take the class, you're going to have access to this one-of-a-kind database where citizen scientists are now sharing this information with each other for in perpetuity, forever. And it's only going to grow the size of the group, because as you teach more classes, you're going to turn people onto this database or whatever, and it will grow. And so it never expires. It doesn't require you to pay a membership to. It's a forever access, correct?
correct. And the part that people are getting full membership to, that does, like, they, like, after a year they could pay for this full membership. All the stuff I mentioned is free. All the things I've said so far is free. But imagine if you could run, like, the analytics like you see on your YouTube or your Facebook, those analytics where they make charts and all those things against any of those variables.
Yeah.
Imagine if you're like we just talked about rhizobia. What would it look like if we were able to measure rhizobia across pH, across bioregions? Whoa. We would instantly see why nitrogen deficiency might be an issue in certain areas, and those areas need to be critically thinking about how they're going to inoculate everything that they can.
Yeah.
Simple things that'll, like, pop out at us, like I want to, like, have people, like, and it's not much money. Well, I'm like, membership for a year would be $45. But the thing is that the people who care, who want to dive in and be the researchers for this, are going to be recognized, and they're going to be able to provide solutions that have never been possible before. And we need to continue to fund it somehow. You know what I mean? And pay for all the information that's going to house, because we need to be able to, like, have the website stably host and then cross-reference all those things. So I need to figure out some way to do that. And the people I've talked to have said that's a really reasonable price, $45 for a year, for the you can measure anything against anything, compare anything against anything, like, and that's including the DNA. You could download the original DNA sequence and rerun it through different databases, because that's what people do now. They're like, which database did you do it through, Matt? And I'm like, the WIMP database, and it's literally called WIMP.
Well,
And people are like, okay, did you do it through NCBI? You know what I mean? Like, or like, did you do it through the German database? And so that ability to allow people to have their hands on the original files and data, I want people to be able to do, then to run it through. And the thing is, people with labs are going to be like, oh my gosh, okay, now I'm going to be able to use this AI that I use for counting microbes to analyze all of this. And they're going to be able to train it. And so they should pay for that service and help fund all of us to have access and all this kind of stuff. But at the same time, that's going to help people. You know what I mean? And all that data is going to be all the uploaders and all the, it's going to be like a library where you literally can search those things and create those data points yourself, like a library. But so the strength is going to be the ease of putting those analytics together, making pie charts and charts, and being able to find things very easily and find those patterns very easily. But I don't want to hold anything back either. I want this to be so incredibly powerful and to grow. That's one of the fundamental things about that you're always doing, Diamond, is research and study. And that means we're going to continuously find new information. And that's science. And so it's exciting, it's thrilling, and these are the tools and these are the skills to explore, discover, document, evaluate, and share the information that you encounter.
Right. So
So
if I let's say I want to go all in on the
yeah.
regenerative soil microscopy course,
Mhm.
how many is it going to be? Weekly, 13-week course? Because like you said, any similar course to this is going to be university level. It's going to cost you thousands of dollars, or it's going to be from some other private permaculturalist who does microscopy. If I get the course, how long is it going to be? And do I have to have any background in chemistry, or can I just go in as a layman?
I'm going to design it so that everyone can start here, and understand what we're doing and why we're doing it. And it's lifetime access, and it's a 16-week course. So we're going to have live Q&A every week. Because if you don't understand it, then you're not getting the value. So I really want to make sure everyone understands it so they can do the work, feel the value, share the value, and then it starts compounding. It's like we're going to discover so much. So I also want people to stay in the community. So it's not like a one and done. They can join any live go through of that 16-week every year experience with new students. And we all know what it's like to be with new students. They're excited, they've got the enthusiasm, and you're like, yeah. And it gets you jazzed up again. But then you've got the experience to see things with new eyes, and it changes it. And that's why I always love to encourage people to just always be part of this. And they ask better questions. It extends the learning for the beginners actually in a unique way, because it creates this, what we call in education, scaffolding, and they can climb up in their comprehension. And so for me, I also get to know people's names. I get to know people as individuals, and they also get recognized. So I've had students in some of my courses come up that way and then start writing their own books, start their own farms, start their own businesses, and it's incredible. But that's what it's all about. It's about facilitating the greatness of everyone involved and allowing for the potential of amazing things to happen. So that's really what it is. I'm giving people the keys to the car, and I'm teaching people to drive in the microscopic world, and the whole world's open for us to explore and connect with. And the reality is we really can assess what's in the soil. You can look under a microscope. You can very quickly see if there's fungal activity. You can very quickly see, if you've got a microscope like this, you can very quickly see if there's if the soil is being if it's alive or dead. Actually, like instantly you can see that. If there's life in it. And then you can see the anaerobic, or like the dead versus living life as well, very easily with a live dead stain. You can see if there's fungi there very, very quickly. Within minutes, you can assess an entire area very easily. You can know how it's cycling. So you know how everyone says, like, this is the mineral soil test I got, and this is what I have. But if you tested your plants, they would have much more than what you saw from all those tests from Texas A&M and other people, because the microbes are actually releasing them, Diamond, from the nutrients in your minerals in your soil.
Yeah.
So your living soil is accessing all the stuff that our tests can't even see. And so that's why it's so, if you see that there's amoebas and you see that there's nematodes, and you know which kind of nematode, you know exactly how things are cycling, and you know that this plant's getting fed. These nutrients are being unlocked. So it's really powerful. But it's part 2 of a whole trilogy. The DNA thing, I got to tell you, I'm going to spill the beans on something just because it's fun, right? Let's do this, right? So when I did the work to do the DNA sequencing on compost, I wasn't expecting to encounter like fundamental truths. And there's so much, like, not just that like EM is in there, but like there's commensal endophytic E. coli that accounts for the majority of all decomposition, all digestion. So it's in all the digestions of all animals already. That's why it's in the manures, but it's inside all the plants to begin with. And then it's fundamental to all decomposition. So commensal mean like we like are cool with it. E. coli is everywhere and all over everything. And there's like 9 to 12 pathogenic E. coli, but there's over like 2 to 3 million different forms of E. coli species.
Let's think about that at the higher level. Okay.
Right. Keep going. Let's go.
We have animals that live on Earth that are pooping everywhere, right?
Yeah.
We have birds that we're trying to bring into our permaculture orchards, and everyone knows it's beneficial, but they just don't know why. Well, we know poop is good for the soil, but in the reality, in the little conversation we've had, what we're now finding is the Earth actually has evolved in a way to supply itself with what it needs on the surface for decomposition in all forests, in all settings. And
Yeah.
it's you eat the plant, comes through your biome and comes out the other end to then hit the earth and then to work for the earth to benefit the decomposition in the subsurface. It's absolutely mind-blowing. And can you imagine with this database what will be revealed?
So we last time talked about EM, and time before that we talked about EM. This is pro EM, and you can eat it. It's USDA approved to eat. In other words, these microbes are inside our gut already. Yeah. And so these are inside, they're plant endophytes as well. So they are the information loop between the soil decomposition in our compost, our plants, and our bodies. This is. And there are more. And the thing is, when we have bad conditions, that there's this thing called horizontal gene transfer, and the microbes absorb the broken up fragments of DNA that comprise more than 40% of everything all around us is covered. More than like this, when we test DNA on the surface of things, we're going to find like 40% unclassified, which is fragments of DNA. DNA theoretically can take millions of years to break down. All microbes are absorbing DNA constantly and exchanging it through conjugation. You know what that is, and then viruses, and so all those pathogens are absorbing genes from the environment to create their type of species. E. coli is like a universal chassis. That's why GMO folk are using it, because it naturally in nature dumps genes and takes them up constantly to adapt to the environment. This is why we're like, the compost microbes just appear out of nowhere. It's amazing. It's like magic. It's not magic at all. It's they're absorbing the DNA from around them and becoming different microbes altogether.
Well,
And they're changing species. They're changing what?
The GMO, the Roundup gene, the GMO gene, the Roundup resistant gene was an accident when a chemist went out to a disgusting toxic waste pond at Monsanto Bayer facility where they were dumping the glyphosate pesticide, and he was taking samples. And guess what he found living in there? E. coli, glyphosate resistant E. coli. And then they replicated it, and then they added it to a seed. And then the seed became resistant to glyphosate. It's that simple. And that was the first GMO.
And it's really, really fascinating that they've demonized that word E. coli. It's like a knee-jerk reaction. And meanwhile,
it is a demon in that case.
Well, yeah, right. Well, they took it and enslaved it. You know, it was actually processing and trying to take care of it and destroy it in the environment. Yeah. And so these truths, these fundamental realities, and the only way to know how good a test is to test it against other tests. And so it's like we're already uncovering. I mean, man, I have hundreds of students already involved in this, and we are discovering so many things. And so I want to open the door. I want to include as many people as possible. And I really appreciate, you sharing this and having me on here, because this is not, not everyone's going to get a microscope, and I know that, but someone, listener, watcher, that
Yeah.
that knows someone, or someone personally that's into soil, that's into compost, that actually wants to know good compost, and actually wants to know good, better, best. Because that's the reality, is we want to be able to actually calibrate this down to a science, so that it's not any more guesswork. There's not any more like false positives running around, because there's a lot of that. If we just don't look, it's okay. Let's fly on.
Now, do you have any good samples of beam?
Of bean.
Beam, BEAM, that the compost that I've been selling for over a year.
Yes, I
I was,
do.
I was going to send you some to
Yeah,
look at.
so that is really, really incredible stuff. If you look at that under the microscope, it's totally fungal, and well, not totally fungal, but it's very fungal compared to most things. And there's spores throughout it. So this is like, if you're like, oh, I already do compost, this is the kind of thing that you could water in to your compost before you apply it, and then it would give it a fungal kick of different biology. And that's the other thing that people don't realize, is that when you combine all these different composts, you make incredible compost. So your home compost, it's awesome, no doubt. But adding in like your neighbor's like vermicompost into that inoculant, adding in bean compost from Diamond all these things create a biological reaction that changes your soil fundamentally. And the
Well,
more actors you have in there, the more potential you have in there. You see it.
yeah,
Those beds that get that
uh,
extra.
I think this could be a huge. I mean, this is a huge wake up call for people out there who poop in buckets. That human manure is the way to go, because if you're eating a clean diet, if you're eating organic, you grow most of your own food or something like that, or you're trying to eat your best. The human manure is going to be a natural resource beyond reproach. We know about the fertilizer shortages that may continue. We have no idea if commercial fertilizer, will be available in the future. But what we do know is that humans will poop forever. And so, yeah, it's that simple. And even
Yeah.
if you have dead poop, if you heat it up to the right temperature for a long enough period of time over a series of years, adding other compost, the end product is going to be clean and brilliant.
I think it's 3 to 6 months. You could use it, but a year is when you have the peak of biology, and it's so funny. I mean, years ago it was like, how fast can you compost? And then there was 18-day Berkeley, and then there was 15-day Elaine Ingham, and it was like, what's next?
12.
15-hour? Ah! And then and now they've got the microwave composter, right? The electric microwave composter. It's like, what? But
Yeah.
the reality is, grandma's compost that sat there for a year or more, or the old farmer's compost that just left the last year's, steer manure or horse manure alone for a year, that those composts just had this incredible biological reaction where it went through anaerobic, and then it went through facultative afterwards, and then the worms worked on it. And so it's
The
natural.
chicken coop we leave on the ground for a year is great, but because it's so dry here, the 2-year-old stuff shrinks by 70 more percent, and then it stays there.
Whoa.
And that stuff I put around, and the
Yeah.
results. Once the water goes through it a few times, and then it's in the subsoil, and then it's late spring. The plants are like it's almost like you injected them with vitamin B12 or something. They are dark green and super vibrant. We have trees that are now 22 feet tall in 5 years.
And so I can pull up on that computer right there. I'd have to find the sheet. I'm in boxes still from the move, but I have a sheet of paper which identifies which of the 6 is the beam compost, because we could literally go to the DNA readout. Because the thing is, I did 6 little samples. I think the total was like maybe 0.1 milliliters of milligrams of soil. And I compared all these different soils. Beam was one of them. 800,000 reads. 800,000 different microbes were detected in those 6 samples.
And what would be a weak sample?
A weak sample out of those individuals would be like 8,000 reads. That would be a weak sample. So that I have basically EM, because it's simplified, is down in those reads. But that makes sense because you're not trying to have everything under the moon. It's not compost, right?
8,000 versus what?
Versus like 200,000.
Yeah. Okay.
So like really good compost is like really diverse, but
well, and again, you were saying old grandma's compost, the Johnson Sioux reactor, that takes 12 to 16 months. Hello.
And the incredible thing about being compost is it combines every single best practice from all the other composts. So it has the aeration, but it's also still, and it has that time frame where it is hot and then it cools and then it stays for a year. So I just think that you can't beat it. And that's why everyone has changed to long format composting.
And for good reason. It's to get
Yeah.
the microbials up. It's to get the all the contents high. Now let's end this now, and let's do a podcast at the middle of the Kickstarter in like 2 weeks after we reach the stretch goal, and let's talk about that beam, and let's get up the microscopy or the data set, and let's do it.
Yeah, no, I'll have the DNA up when we do it next time, and I'll have it behind me on the microscope, and we'll like pull the screen and like put it right next to it so you can see it.
Yeah.
Because the reality is, I mean, microscopy is not hard. It's literally what Diamond said. You shake things up, or you put, seeds in little things like this. This is a seed that we're going to look at once it sprouts. It's in EM. And you put it under the microscope. This was a root with no cover slip on it. I just threw it under there. So this is really easy. You're taking a drop and putting another piece of glass on top of another piece of glass, or you're just putting it under there naked and just looking at it. It's understanding what you're looking at, understanding which is the kind of microscope that's really powerful, and being and then being part of this incredible community that is going to spread the education, fluency, documentation, connection, caveats, conditions, all the limitations and advantages, comparison over time so you can track your progress, comparison to your bioregion so you can know how you are in comparison to everyone else. It's going to change everything. And so I invite everyone to take part in this regenerative soil microscopy. Show your friends that are into compost and soil. You know who they are. because this is going to be right up their alley, and this is going to allow them to take action on all of that rather than just, building compost and hoping that it's the right thing. This is going to allow them to know.
Yeah, and I'm sure Matt wouldn't be upset if you and your friends all chipped in together and bought the course together and didn't say anything. I mean, because a lot of people can't afford it, right? But you would be happy if 5 people could get together and pay for your course and all watch it in the basement.
La,
But
la,
they only,
la, la, la.
they only get one spot in the database, unfortunately. So,
But yeah, no, I mean, even like the book, like that's the thing. You don't need to like be like in the course to add to the database. I'm not trying to like, control. I want like everyone. It's an open database that anyone can add to.
So if you
And the
have this book and you have your own microscope, you can add to the database for $45 a month or a year.
Well, if you want all those analytics, you can just add. I just want to set it up so that you can add to it and be part of it, regardless of being able to pay or not.
Oh, so with the payment, then you can do the analytics.
Yeah, because
Oh,
the people who want that information are already paying top dollar for other things in their setup.
yeah.
Does that make sense? Like they've
Yeah,
already invested if they care about that. And so I'm like, those are the kind of people that are going to help all the, like everyone else, get access to this information. Does that make sense?
beautifully.
All right. Yeah. Cause I figured it out. I was like, I got to make this as open as possible and empowering as possible. But at the same time, I got to figure out how to pay for it. And so it's those deep analytics, comparing the DNA to the biology, being able to download those original files, run them through, like, because the thing is, there's open AI that lets you count the bacteria on a slide
Yeah,
already.
yeah, yeah.
We just need someone who cares enough to spend the time to do a few other things that'll pair with what we're about to do. And the breakthroughs are going to just blow everyone's mind. So let's just start the process. Join us. It's going to be fun.
Yeah, and the textbook's almost ready. He still has to do all the photos. I mean, the manuscript is written. So for $100, you can get a textbook that I can tell you the last textbook was worth $100 in my opinion. But I only think how much is I don't know how much the text is, 50, 80 bucks, whatever. The book itself is worth $100. Regenerative soil microscopy. And I think for that value, you're going to sign all the initial copies, won't you?
For that value, they get into the database at for an extra, so everyone beta tests it with me, because I'm not going to charge for something or like use their free year up on beta testing.
No.
So I want to do like a beta test version and have everyone give me feedback, and then I'll begin the year. So the year would probably begin next February. And then, so they'd get like a year and a half access to all the deep analytics. And then they would still be able to upload and participate. And
And the first
I
edition,
really want people to get excited and get involved. Like
these
that's
first editions are limited printings. Like the first edition of Regenerative Soil. I mean, how many copies was the first printing?
the first edition was only 1,000 copies. 1,500 copies that were printed.
And how many copies?
And those were all signed at the Kickstarter and sent out, and there are no more of them. They're
Yes, that's
gone.
the whole point here. That this is going to be the same, and you have an opportunity to own. I mean, these aren't just textbooks. These are
Visual guides.
visual. I think they're art pieces because they're beautiful as well as representative and very stimulating to and very peaceful. I mean, these are beautiful works of art and science. And so for $100, you get the book and you get a half a year of the beta and then a full year of the data going up. That sounds like a boom to me.
You know, this is something that became like evident to me when all right, so in Germany there's 2 different certified soil labs, and I'm friends with one of them. One of them is one of my students as well. And they had a savvy farmer give them the same sample, and they gave back different answers. And they're
Oh,
professional. They're charging hundreds of dollars for these things.
I heard.
And they literally went on Zoom and then side by side did it together and then still got different answers, like totally different answers. And so the recommendations were divergent to the farmer. And so it was terribly embarrassing. But that's the type 1 error that we face if we don't create a real open, transparent database. And we start talking about these things and showing our work. So that's what this is really about. This is about opening the door to truth or the path of truth, you know?
Happening around the world. Big Ag is taking control of the food supply. And now Matt Powers is taking that back. And you could join the movement and become a member of Regenerative Soil Microscopy and that database. And you don't even have to get the book. You don't have to buy the course. You can just be part of the database for a $45 donation to the Kickstarter. Matt, it's always a pleasure. You're almost there. You're 1/5 of the way there in just about 10 hours.
Thank you.
That in 2 weeks we'll be at a stretch goal, and hopefully it involves a tent and a trip to Tahiti, but probably not. So we'll see.
I was thinking an app.
Oh, yeah.
Turning into an app. that we can upload, so we can take the pictures, upload everything on a
Soil,
phone.
it tells you what's in it.
It's gonna be amazing.
All right, let's do it. So, guys, help out Matt and his Kickstarter. It is fantastic. What is the name of it again? Regenerative Soil Microscopy, the book, the course, the database, and that is a boom to Matt Powers. We're going to have him on in 2 weeks, where we are going to reveal to you what's in Beam and why it is the bomb.
Yes.
Matt, always a pleasure. We love you.
Thank you so much.
Yeah, and good luck.
I'll see you in 2 weeks.
Yeah,
Thank you, my friend.
Do it as soon as you hit the goal. So if it's that's in 7 days, I'll be calling you up.
Okay. Yeah, absolutely. Thank you so much.
Yeah, thank you. We love you.
Have a good night, everyone.